Hello, I would like to briefly introduce to you human metabolome technologies. HMT was established in 2003 in collaboration with Keio University with over 10 years experience. Our platform is based on capillary electrophoresis mass spectrometry, and we have developed a proprietary data analysis program. We have a large library of over 1,200 compounds, where 900 are annotated and over 100 are accurately quantitatable. We can provide complex statistical analyses, and we provide you with a comprehensive pathway analysis. Our technology is based on capillary electrophoresis mass spec, whereby compounds are first separated based on their charge and molecular size. In the electric field generated by applying a voltage to both ends of the capillary, the direction and rate of migration of ions depends on the physical properties of the metabolites. In this schematic, we are separating the positive compounds, but a reversal of the charge applied to the capillary will separate the anionic compounds. The compounds are then separated based on their mass and time in time of flight or QQQ-MS. Concurrent separation of hundreds of metabolites present in assay samples is achieved through these differences in mobility of ions. There are several advantages to adding a CE-MS platform for metabolite profiling, which include higher resolution and sensitivity. CE can identify a unique set of metabolites compared to either LC or GC-MS. CE-MS has a fast analysis time with improved reproducibility with a low RSD. CE analysis includes data reduction using our proprietary software tools. Here you can see the low RSD achieved as a result of measuring a 100 micromolar cationic solution 6 times. Typically, RSDs are at 2% or below. Similarly, low RSDs are achieved in anion measurement mode, again with most RSDs at 2% or below. No single platform is capable of measuring the entire metabolome. CE-MS is best suited for the detection of the majority of small polar metabolites, though unable to detect the more non-polar steroids and fatty acids. However, it is clear that CE-MS is capable of detecting the majority of the metabolites involved in cellular metabolism. In particular, the TCA and urea cycle intermediates, phosphosugars of glycolysis, and the pentose phosphates. In addition, though LC-MS has methods for detecting nucleotides, these compounds are particularly suited for analysis by CE. Here we can see a nucleotide comparative study performed using CE and LC-MS, and it was shown that cleaner separation and better peak shape were achieved using CE-MS, as well as increased detection sensitivity, which leads to better quantitation, accuracy, and reproducibility. In CE, the mobility of ions is not only dependent on their charge, but also their radius, which allows us to effectively separate structural isomers of the same mass, but with different dimensions. An example is our ability to separate glucose 1-phosphate from fructose 6-phosphate and glucose 6-phosphate, which all have the same mass-to-charge ratio. Most LC-MS platforms detect these 3 metabolites in a single peak, and thus report these as combined M over Z values. However, these 3 metabolites can be involved in different metabolic processes, for example, glycolysis versus glycogen synthesis and degradation pathways. HMT has a number of analysis packages that are tailored for biomarker discovery, basic profiling, and lipid analysis. We have our basic scan, which is an untargeted analysis approach, which utilizes our annotation list of over 900 compounds. In addition, the advanced scan option, which is best suited for biomarker discovery, not only reports peaks from our annotation list, but also reports unknown compounds that were detected. Our dual scan combines our CE technology with LC-MS to report both the polar compounds as well as fatty acids and steroids. Our CARCINOSCOPE package is our targeted quantitative analysis option. CARCINOSCOPE provides the accurate concentrations of 116 metabolites involved in primary energy metabolism, and because of this quantitation, it also evaluates 30 metabolic parameters for facilitating data interpretation, such as energy charge, lactate to pyruvate ratio, glutathione redox ratio, and total amino acids. CARCINOSCOPE combines CE with both time of flight and triple Q mass spec, which allows for higher detection sensitivity and analytical accuracy. Full statistical analyses are provided, which includes PCA analysis and hierarchical cluster analysis, along with a comprehensive pathway analysis. CARCINOSCOPE is particularly suited for central cellular metabolism. This includes glycolysis and the TCA cycle, but also the pentose phosphate pathway and glycogen metabolism. Amino acid metabolism is also central to a cell's ability to synthesize de novo proteins and nucleotides, and can be indicators of muscle wasting or protein degradation. Our analysis also includes the urea cycle. Additional metabolic analyses include the evaluation of oxidative stress through the glutathione redox ratio and the evaluation of energy balance via total purine phosphates. Biomarkers, such as 2-hydroxyglutarate, the accumulation of which results from isocitrate dehydrogenase mutations, among others, can act as biomarkers of metabolic enzyme abnormalities. These metabolic analyses can strengthen your traditional genomic and proteomic analyses. CARCINOSCOPE provides all at once, quantitatively. As you can see, the CARCINOSCOPE analysis covers the main areas of energy metabolism. Here is a typical example of the data presented as a poster map. Each graph represents an individual metabolite superimposed within its metabolic network. This way, the relationships among metabolites can be better elucidated in the context of its particular pathway. As I mentioned, a very unique feature of our CARCINOSCOPE analysis package is the ability to accurately quantify 116 metabolites, and thus all of the data presented has specific units, in this case nanomolar per gram tissue. This allows for inter-study comparisons performed at different times, as well as comparisons between different metabolites. In addition, because we can quantitate these metabolites, a number of metabolic parameters can be analyzed that covers a range of processes from oxidative stress and viability to amino acid content and DNA methylation potential. The analysis report of CARCINOSCOPE also includes results of several multivariate analyses, such as hierarchical clustering analysis and principal component analysis, which generate a heat map representation of the data and a PCA score plot respectively. Both of these figures are helpful for comprehending the holistic trends of the metabolomic profiles of each sample and sample group, and the figures are also ready to be used for journal articles or presentation slides. The raw data, along with averages, standard deviations, and p-values are also provided, and therefore users can easily check the original data or further modify the data for different statistical analyses. Here we have a study example that examined a range of lung and prostate tumor types. Initially, we can see differences between the normal and tumor tissue. However, using the PCA analysis, it was found that while the lung samples tended to cluster based on tumor type, being either adenocarcinoma or squamous cell carcinoma, the prostate samples tended to cluster based on differentiation status, which can be useful for linking metabolism to tumor stratification. Additionally, we have our untargeted analysis packages specifically geared towards biomarker discovery. In our advanced scan package, we report the known and unknown metabolites detected, which is particularly useful for biomarker identification and patient stratification. HMT has a number of ongoing studies, of which some have led to the development of novel diagnostic markers. In particular, we have developed a biomarker as a diagnostic tool for major depressive disorder. In a clinical study, healthy subjects were compared to both MDD patients and adjustment disorder patients, as determined by their CESD scores, which are given based on psychological evaluations, which can be highly subjective and variable. In this study, plasma samples were analyzed with 538 metabolites identified, of which 193 were annotated. Among the three groups, a single metabolite, ethanoamine phosphate, was identified that could differentiate the major depressive individuals from the others. EAP was significantly reduced relative to the non-MDD control group, as well as significantly reduced compared to the adjustment disorder group. HMT has extensive experience with a large number of sample types and can provide optimized protocols for your sample of interest. We consistently analyze biofluids such as serum and urine, as well as tissues such as liver and stomach. We have specific protocols for both adherent and floating cell cultures, as well as for bacterial growths. Our sample requirement is minimal, which is convenient for the in vitro and in vivo researcher. We require approximately 50 to 100 microliters of fluids, 30 to 50 milligrams of tissue, and about 1 million cells. You can see that HMT has an integrated process where we work with you as a client using our experience with experimental design and sample processing. Our proprietary software is specifically designed to extract the most information that is valuable to you, and our metabolism experts assist you in the data analysis and interpretation. Thank you for your time, and we hope we can work with you in the future.